Hello everyone, I welcome you to my channel where we are going to talk everything about biology related to biology. So we are starting with our first lecture that is going to be a very simple introductory session where we are talking about cells. As we already know that cells are the basic unit of life. We actually started our life as a single cell, right? So what are going to be the learning outcomes in this chapter? We are going to see the overview. We'll see why cell is called as a basic unit of life. We're going to talk about various organelles and majorly the plasma membrane, right? Now, if we see here what actually cell theory is. So I am hoping that the base is already clear at your part. If not, we will be doing it in detail. Actually, the cell theory came up with the point that all cells arise from pre-existing cells, right? All cells come from pre-existing cells only. Then there is a term which says about that all cells come from pre-existing cells. Then second point, that all organisms are composed of, right? All organisms are composed of cells. Now, if I see here, if I tell you the level of organization here, just to quickly revise it. So we already know that cells, they combine together to make up the tissues. The tissues combine together to make up the organs. The organs make up the organ system. And these organ systems actually combine together to make up the organism, like we ourselves. We are the complete organisms. We have various organ systems in our body which are carrying out the various functions, right? So we have respiratory system taking care of all the gaseous exchange in your body. Then we have our circulatory system which is taking care of all the transportation in the blood, whether we are talking about the transportation of hormones, nutrients, and all of that stuff, right? Whenever we say that all cells arise from pre-existing cells, when we say all organisms are composed of cells, there is one more point which cell theory talks about. Very, very important. That is, cell is the smallest living organizational unit, as we can see it here. It is the smallest living organizational unit, right? So here you can see this picture, which is actually talking about the biogenesis, right? It is talking about the biogenesis. What do you mean by biogenesis? So when we say bio, we mean life. When we say genesis, we mean formation. So how actually the formation of life actually took place, that we are going to talk about with this example shown. This diagram is actually showing you how the Pasteur's experiment disproved the theory of spontaneous generation before. So basically, what biogenesis means? Biogenesis means that cells arise from pre-existing cells. For example, the liver cell is going to be developed from the liver cell itself, right? So there was actually the spontaneous generation which was accepted as the origin of small organisms, such as the maggots, right? In other words, we can say that maggots would suddenly be formed from anything, right? But later on in year 1859, what happens? The Louis Pasteur, he disproved the theory of spontaneous generation. But what he did, as you can see in the diagram, he has taken a broth which is being boiled in a swan neck, right? The shape of the flask is like that of the swan neck shape they are talking about, right? Later on, what they have seen, as the time passes, the broth remains free of contamination by microorganisms. There was no contamination being seen, right? There was no contamination being seen. What happens later on? The swan neck thing was removed from the flask. The swan neck was removed from the flask, and the Pasteur actually did that. He eventually seen that consequently microorganisms started contaminating the broth, right? So each flask was having the swan neck to prevent the contamination in the air so that it should not reach the broth. Now, at this point, no microorganisms grew in either of the flasks, right? But when the swan neck was broken on one flask and the broth was exposed to air, so air is having the moisture, the microorganisms they started to grow in it. But the unbroken flask, it remained free of microorganisms. So in the unbroken flask, you can see there is no growth of the microorganisms, but in the broken part, you can see there is a growth of micro, right? As you can see, here we can see the growth, but in this part, we cannot see any microorganisms growing, right? I hope that's fine. They are showing the growth of microorganisms, which actually took place because of the moisture. because of air exposure, right? This is all we have done. You can just quickly have a look what we are actually discussing, and then we'll go on to the next. I'm going to take you to a small, just a quick brief about the cell theory. Who actually talked about the cell theory? Let's see the picture, one of the picture here. I'll be adding it, and we will revise it together. Let's maximize it a little bit. Let's come down. And here we are actually going to talk about the history of cell discovery, right? Actually, the history of precise. Now, in this first scientist which came up in our mind was the Robert Hooke. He actually talked about, he actually was the one who gave the term cell, right? He gave the term cell, and he used his own made microscope to talk about it. So his drawing of his light microscope in Micrographia, which was actually published. Micrographia is the book in which his work was published in the year 1665. This is important. We need to remember that, right? Now, what he did actually, he actually took a thin slice of cork from the bark of the tree. So, what are the keywords we need to remember here? He took the thin slice of cork, right, from the bark of tree. That's easy to understand also, right? And then what he did, he examined it under the microscope, which he actually made by himself. He actually seen that that bark was made of hundreds of little empty boxes, but hundreds of little empty boxes were looking like and giving appearance of honeycomb appearance. So I'll just draw this here. Have a look. It was something like this. These were present like this. These were small compartments, and those small compartments were given the name as cells by the Robert Hooke. Each compartment he saw in the bark was giving the appearance of honeycomb, right? So this is how he actually began with the history of cell theory. Robert Hooke major contribution, but we need to take care that he actually did the discovery of a dead cell, not the living cell. But then who came and who did the discovery for your living cell? That was another scientist. Let's talk about the next scientist, which is nothing, no one but yes, your own Leeuwenhoek. He was the one who observed the living cells, right? He included the bacteria, the blood cells, and the sperm. So I'll just write it here. Second scientist contributed in the cell theory was Leeuwenhoek. He talked about living cells. He was the first to observe the living cells. Which cells he observed? He observed the bacteria, the sperm, which is the male germ cell, right, or the male gamete, and the RBCs, or your red blood cells, right? So I am hoping that till here everything is all good. See, if you have any doubts in the between, you can actually write it in the comment section. How you are able to see and understand and all this stuff, you can actually mention it in the comment section, right? Let's move on with the next part. It's going to be about the third scientist, whose name is eventually the Lamarck and Dutroux. They actually tell that all living things are composed of cells, right? Jean-Baptiste Lamarck stated that living things are a mass of cells and they have their complex solutions move in and out of the cell. So basically, we can understand one point here that next came up the Skledin and Swan, which told us that cells are organized into tissues, and eventually the Skledin and Swan gave the cell theory. The Skledin worked on the plant cells, Swan worked on the animal cells, and eventually they gave up the cell theory. So I just write the scientists' name here. They are Skledin and then Swan. The cell theory was further modified by the Virchow. Right? That's the name of the scientist, Virchow. Now, what did he said? He said that since till now it was already clear that this Robert Virchow discovered that two cells were formed by single cell dividing into two, and his discovery, the Rudolf Virchow discovery, popularized the theory of biogenesis, that all cells comprise from or the come from pre-existing cells. Since the virtual was getting too much of popularity, this theory was easily acceptable in the Europe and then obviously the rest of the world. Now we learn it in our science books, in our biology books, whether you are talking about CBSE curriculum, NEET curriculum, wherever you are, where to talk about that. Just see the picture, observe it. These are the important names. Remember, I write the terms here. Important one. This Sledden worked on the plant cell, and the swan worked on the animal cell. Right? These are the basics we need to keep in mind. That what all about the cells we are talking. You just have a look here. We just learned about we have just taken and seen the overview about the cells. Cells is the basic unit of life, right? And that's how the scientists have contributed in the discovery of the cell here. That's all we have done till now. Now let's see that what are the common cell features we are able to in the various structures of cell, whether we are talking about a plant cell, I'm talking about the animal cell, or I'm talking about a simple bacteria. So what are the common stuff which we see? So let me just show you draw a simple diagram here to make you understand that when I'm talking about a cell, in general, what cell is actually having. For example, this is the cell I have drawn, right? So let's just make it a little more clear. You can also take the shapes. Yeah, this is the cell for say. And now the outer membrane you are seeing here, that's going to be the plasma membrane, which allows the entry and exit of everything apart in and out of the cell. Inside we have the structure which is called as nucleus, which actually controls the every function, control overall function of cell, right? Then we have this fluid material in the cell, which is known as cytoplasm, right? These are the basic things which we need. You can see in cell structure is, to be precise, there are three structures which I am talking about here. That is your number one is your plasma membrane, right? Then your nucleus, and then your cytoplasm. So we use plasma membrane as cell membrane. We also call it as cellular cell lemma or the plasma lemma, the same thing. These are the basic things which we see in every cell we study about, whether it's a plant cell, animal cell, or the bacteria, right? So if we go a little above, I will just put a simple picture here to make you understand and observe basic structures in plant, animal, and so whatever pictures I am sharing here, they are specifically from the uh, publications of the high school students who are in year 11 and year 12 under the VCE curriculum. This book which I am having, the Henman units 1 and 2 I am following over here. So if you are targeting the unit 1 and 2, you can study with me and follow my channel for further updates on the same topic. We will be doing the units in very detail, topic wise. We are going to cover every part. So yeah, this is going to be the part for our next part of our session where I am showing you the pictures where you can see the observations of how you can see the cytoplasm, right? The cell wall is present definitely in the plant cells. We have the nucleus, which is containing your DNA, the vacuole. We can see this vacuole is actually comprising most of the space in the plant cell, right? This is comprising most of the space in the plant cell, which I am using also. Then you can see this is the animal cell, the B part, which is again having the plasma membrane, cytoplasm, nucleus, which is having the chromosomal DNA. The ribosomes are also present over here, right? Then this is the bacteria, which is having outer covering as a vacuole. It also have a cell wall, which is also present in the plants. But obviously, animals do not have cell wall because we don't need it. Plasma membrane is present. Ribosome again are present. Nucleoid, circular DNA, which is not, it is non-membrane. Basically, it is not bound by any other membrane. We have the flagella. They are required for the movement of the bacteria. Apart from flagella, the bacteria are also having villi. They are also membranes, which are helping them in their further movement, right? We also have the cytoplasm, which is comprising of the fluid material inside it. This is just an overview of how cells are being arranged, having the different structures. See, there is another of all the different names or the term mentioned over here. There's the mitochondria in the animal cell, but there's the chloroplast in the plant cell, right? I'll be telling you, and I'll be writing the quick key terms over here. Plant cell. This one is animal cell and bacteria. I hope what all we have discussed here is all clear to you. You can actually pause it, take the snips, take the screenshot, make your notes while listening to me. And this is how we are going to take up the sessions in the later part so that everything is more clear to you in a simple format. I will be meeting you in the next session. I hope you are doing well in your studies. Bye bye. Take care.